I'll start with the first question. There are few which were asked during the class, but I mean, just like confirmation. So basically, the first question is: are green bee-eater saline species?
Yeah, that's a great observation. Yes, you often see green bee-eaters perched on good vantage points and are scanning the area for insects. And they swoop down to catch bees and other insects that are flying about. Yes, they are great examples of saline species.
Okay, and this question might have been answered during the course of the lecture, but I'll still ask it off. I mean, apart from drongos, what are the other common sentinel species?
Right. So drongos mostly drongos are seem to be sentinel species in mixed species flocks that we commonly see here in the western parts of India. In other flocking systems, there are other tanagers, for example, in the neotropics that are sentinel species. Even among the intraspecifically gregarious species, some individuals tend to be sentinels, right? So, for example, if you've seen flocks of jungle babblers, there will often be a single individual that will choose a higher vantage point and scan the area. So, even groups of intraspecifically gregarious species are known to have sentinels within them. Other species that are saline species, like paradise flycatcher, monarchs, they also have similar feeding habit, except these are largely understory birds. They are known to make alarm calls. You will see them giving out alarm calls every now and then, but they do not perch higher up like the drongos, and they're of course not as bold as the drongos, but they are saline species also. They may not always act as sentinels. So yeah, interspecifically gregarious species also have sentinels and drongos, and there are a few other examples, like I said, from the neotropics, the tanagers are sentinel species.
All right. And the next question is there a sex-specific differentiation between a sentinel in mixed flock? Like, is there a particular male or a female bird of a particular species that act as sentinel?
Right. That's an interesting one. As far as I know, this has not been looked into. So this is especially difficult to study in species where there is very little dimorphism, right? And in drongos, at least, it's not very easy to tell apart male and females just visually. I do not know whether there is a sex-specific bias in what acts as a sentinel and what does not. It might be an interesting study.
Thank you, Priti. So the next question is, so basically the question is asking, why do the specifically gregarious species lead? Like, what is the reason behind them leading the group?
Yeah, I'm trying to read the question. Why do these interspecifically gregarious species lead? What is the reason? As they are in a group, do they also give cover to the flock to move safely? Okay. So interspecifically gregarious species are social species that are also, like I said, they may also be in their own family groups. As far as to why they lead, one can only speculate. If you want to answer why questions, you may have to do experimental manipulation. But interspecifically gregarious species generally, when they are seen to sort of cross over a particular patch or a particular open patch or a particular stream or path, you do see them going back and forth. Okay, some birds will fly across the stream and then come back, and then some more will pass. So we don't know whether they give cover to the flock, but they do seem to be found in the forefront of the flock. Why they do that is a question that I don't think has a very clear answer yet. There could be several reasons. So if the species are family group, then and they are cooperatively breeding, then there may be some kin benefit to that particular behavior, and as a result of that particular sort of evolutionary hangover, those benefits are also reaped by the species that are following them. So that could be one reason why they seem to be in the leadership position. So there is some amount of cooperation in the family group itself, which then benefits the rest of the species that also follow them, which is probably what makes them great candidates to be leader species. And that's also related to another behavior of gregarious species, which is early alarm calling. So if you look at cooperatively breeding birds, they are known to have really well-developed alarm calling systems. So intraspecifically gregarious species, because they are cooperatively breeders, tend to be alarm callers, so early alarm callers. And that also makes them great as flock leaders.
Okay. So the next, so Krishna is asking for more elaboration on nuclear species characteristics.
Okay, sure. So I would say that don't get too caught up in the terminology of nuclear species itself. What people I will still go ahead and describe to you what people have described as nuclear species. Nuclear species tend to be those that are important in flock initiation, and they're also important in making sure that the flock stays together, and they're also important in what we are calling leadership, right? What they flock moves together. The species are followed by just specifically gregarious species or nuclear species are followed by other species. These tend to be species that are more conspicuous in terms of numbers, that they will be several in numbers and will be easy to detect. They may act as cues for mixed species flocks. So if there is some activity there, you may be able to guess that there is a mixed flock there, and curious birds might go and check out that particular mixed flock if they want to join or not. They also tend to be noisy. They tend to make more alarm vocalizations. They tend to make contact calls as well. Contact calls are calls that species make to they're sort of like check-in calls, right? All okay kind of calls. You may have seen jungle babbler groups if you go bird watching often, that there is a lot of calling, like chup chup calls that species make to stay in. It seems people have described contact calls as all okay calls, right? So nuclear species also tend to have these interesting contact calls. They, in some cases, have been described as also being behaviorally conspicuous. So they are so fidgety and they're so flitty that other species tend to follow them, and also because they then tend to flush out so many insects. So all of these in combination, you can see now already clearly that there's no one single definition of nuclearity. Right? There's no one particular line of definition that I can say that this clearly defines a nuclear species. So there are several different factors that play into this, and which is what makes it really difficult to define. And because of that, I refrain from using the term nuclear so often in the talk, except for that one slide, because you're very likely to encounter it everywhere in the literature. And hence I call them some species that are important in flocks, one of them being sentinel or saling species, the others being intraspecifically gregarious species. But it's a combination of all of these factors that make species nuclear.
Thank you, Priti. So the next is again an elaboration on the phrase that you use, copying locations.
Okay. Well, by copying location, I mainly mean that if a particular individual has discovered food, I might be able to go and take advantage of that. So it's food that's already found. So you can go and copy that foraging location and benefit from. You don't have to do your own searching, or your searching effort is then considerably reduced. That's what I meant by copying.
All right. Krishna is asking, probably she's asking what flushed insects are, and do birds eat only specific insects while the others are flushed? So she wants an example as well.
Okay. What I meant by flushed insects is that insects that are disturbed by the movement of other birds. If you have observed I don't know what group of maybe babblers feeding, they often turn to turn leaf litter. They are very they are very active, that may lead to insects being flushed out or being stirred up from the ground, from the canopy, from the leaves in the trees, and so on. That's what I meant by flushed insects. They're not a particular kind of insects, but they are insects that are disturbed by activities of other birds. That's what I meant by flushed insects. Do birds eat only specific insects and others are flushed? So I'm not entirely sure what you mean by this question, but flushed insects are eaten by birds that will sally. So if there's a drongo that is following a bunch of warblers, then these birds, the insects that get disturbed by their activity is often eaten by, are caught by the drongo. And specificity about what insects get eaten is, it's a little bit challenging to study. So I don't think there's been very specific work on what insects some of these birds are eating.
Okay.
I hope it's clear.
Yeah. So the next question is kind of interesting. See, are leaders leading the flock, or are they just being followed by other birds?
Excellent. Yeah, that is a great that is a great question. Yeah, leaders may be leaders because they're being followed, right? The others are not. They're not necessarily leading the flock, but the others are staying behind those. So yeah, both are possibilities. You would have to do some experimental work to be able to answer this. But if you look at some of the flocking literature, you see that definition of leaders are such that they lead or are followed. So people have used both to describe leader species.
All right. I think I
also saw Umesh join in. So in case Umesh wants to, are you there, Umesh?
I think he's there for the 4 o'clock session.
Okay.
Where you both answer the question. Yeah.
Right. Okay.
So yeah. So.
Sorry, he was not added as a co-host, so I just made him the co-host.
Okay. I was just saying that if Umesh has also studied mixed species flocks, and if he has anything to contribute to some of these answers, please feel free to pitch in, Umesh.
Okay, so moving on to the next question. Parth is asking, what benefits do the intra-specifically gregarious species get from joining the flock?
Right. So that's a good question. If we are talking about them largely as birds that are leading and the other birds are benefiting, then what are gregarious species getting from being in the flock themselves, right? So some of the protection benefits and some of the foraging benefits are common to all species that participate in flocks, right? So the improved protection from predators is a benefit that all species get. So the gregarious species are also benefiting from increased group size of the group if there are other bird species that join the flocks. If there's an overall improvement in foraging, in food finding, the interspecifically gregarious species are also benefiting from that overall increased food foraging efficiency in flocks.
Okay, so Krishna is asking if there are more than one canopy species in mixed species flocks, and if there are, then aren't they competing, and how is it that they're able to coexist?
Right.
An example.
Right. Yeah. Great question. Yes, there are often more than one canopy species, or often more than one species that feed in one particular forest strata in mixed species flocks. For example, you'll see ashidrongos and minivets that largely forage in the canopy, and they coexist. Yeah, they sure do coexist. And how do they then coexist is the question. They have slightly different feeding habits. First of all, insects are a dispersed resource, right? They're not a clumped resource like you would imagine a fruiting tree to be or a flowering bush to be. And insects, in a tropical context, insects are also a fairly abundant resource. Secondly, an ashidrongo and a minivet are both canopy species, but they forage differently. While a drongo may be a sallying species that catches insects mostly while sallying mid-air, Minivids are species that are sally gleaners, that sally out, but also tend to pick insects off of surfaces of leaves. So there's a little bit of partitioning there, right? That's what I was trying to talk about towards the end of my lecture, that you may be similar in certain aspects, which is here that you're using the same strata of the canopy, but you're different in other aspects. Here, the example was that they're different in foraging technique that they use, or the foraging maneuver that they use when catching an insect. That is how they're able to coexist.
Okay. Next to us, like, would a flock of common and pied mynas or a flock of chestnut-tailed starlings and rosy starlings be called a mixed species flock?
Right. So sure, there are multiple species and they are moving together. So you would think that it is a mixed species flock, right? So in the classical sense, in what we were talking about, these forest birds feeding in mixed species flocks, maybe the mynas and the starlings don't typically fit that definition. But that's not to say that there are not more than two species. Here, when I was defining, or when I was talking about what a mixed species flock is, we spoke about a few different things, right? These are moving groups of forest birds, insectivorous birds, mostly passerine birds that are feeding together, that are moving together, that stay together for a given amount of time, that navigate the forest and stay in the canopy and understory while food finding, while foraging. So that would be a typical forest birds flock. A flock of myna, starlings, we would have to look at what the context is. Are they external stimulus? Is there an external stimulus? Is there an external resource that has attracted these starlings and mynas to that particular area that you're looking at? Or are these only groups that are flying about, right? You often see starling groups that take off, and there you also see rosy starling murmurations, which everybody has seen. So those contexts are slightly different from what we were talking about today. I'm not saying that those are not mixed groups, but they are not like the groups that we were speaking about in today's lecture.
Okay, so Aditi was asking how, or basically asking you to elaborate on the mate choice as a benefit of mixed species flock.
Oh, no. Yeah, Aditi, sorry if that was confusing. And I did not say that mate choice was a benefit of mixed species flock. I was talking about groups in general, and that leks, which are aggregations of females, of males of the same species which congregate in a particular area, and females come to these leks for these to check out these displaying males. That was the mating context that I was talking about. It was not in the context of mixed species flocks.
Okay. So there was this question about lecture being available later. I think it was recorded. So yeah, that should answer that question. And the next is, are these flocks randomly formed, or are there specific individuals that regularly come together for foraging?
Right. Thanks. That's a great question. I'm not exactly sure what you mean by randomly formed, but I do understand the later part of your question, which I think explains the question better. So there are studies that have been done on banded birds, where you know individual identities of birds, and it has been seen that these studies have been done in Australia in some birds. In Australia on some birds, there have been studies from the neotropics as well on banded individuals, and you are right in guessing that there are the same individuals that come together and associate with each other in mixed flocks in these areas, day after day, and stay together for a long time. One thing that I forgot to mention during the course of the lecture was also that some neotropical flocks are also permanent flocks, in the sense that they associate for really long durations throughout the day. They're almost together all the time. And flocks also hold territories. So mixed flocks, they are in a particular area, they'll stay together, they'll roost in areas that are not too far from where the flock was foraging, and then come back the next morning. So yeah, species do, or individuals do tend to associate with the same individuals day after day. But having said that, in India, there are no studies on tagged individuals, individuals that have where we've been able to identify the identity of an individual bird. But if you do go if you are doing field work in some parts of India, you may see that if you visit a particular part of the forest time after time after time, you are likely to see flocks that have similar compositions at the same spot, which makes you wonder whether they are the same birds. But there will have to be studies on tagged individuals to be able to say anything certainly about that. In other parts of the world, there is more evidence that same species, same individuals associate. Again.
Okay. So Sulochana is asking, since drongos and woodpeckers forage without being a part of a flock also, so is there a trigger? Like, is it when other species are nearby that they join or recruit themselves into the flock?
Yeah, that's a good observation. That it's not to say that woodpeckers and drongos don't forage outside the flock. Typically, you will see flocks being formed in sort of late in the morning. So you avoid early morning hours when the flocks are still forming. So you won't see flocking activity so much early on during the day. As the day progresses and the sun comes up, the day gets slightly hotter, you'll see flocking activity beginning. As for specific triggers, I'm not aware of anything really specific that triggers flocking. It could be that the bird is over here, a particular group of feeding, or there's a sudden vocalization activity which stimulates sort of flocking behavior. But I don't think there has been any study on what triggers flocking. There have been playback experiments of the kind that I described during the course of my talk on certain species, so like the babbler drongo playback that I described. So such vocalizations do tend to attract other species. So perhaps vocalizations may play an important role in initiation of flocks. But you're right that you do see drongos forage by themselves, you do see woodpeckers forage by themselves. Yeah, but I'm not aware of specific triggers. Umesh, do you have something to add to this?
Okay.
Yeah, maybe not.
Yeah, I think. Okay, so we'll move on to the next question. So Parth is asking if, I don't know if it's a hypothesis, but many eyes hypothesis and early warning hypothesis, are they two separate mechanisms, or should they be like merged together as one?
That's interesting to think about, like theoretically to think about. So yeah, many eyes would lead to early detection is I think what he's trying to see. Yeah, perhaps you could think of it as related to each other, but there may be some, so many eyes also just means that there are more eyes looking out for predators, and early warning could also come from specific species that are good at detecting predators, which for example the drongo or some other saline species which are better at detecting. So early warning doesn't necessarily have to doesn't have to follow from many eyes, but you're right that it may follow from many eyes. That's an interesting point. Thanks.
Okay, I think Chandrasekharan is trying to ask for an example of obligate flock foragers in India, probably, or somewhere else.
Right. So it's a little bit hard to say, give specific examples of obligate flock foragers, but there are some species that are always followed. So in winters, like for example, I went in the place that I studied in Anchi, in northern Karnataka. I used to try and find birds that are interspecifically gregarious species that are that forage by themselves, that are in single species groups. But what I found was that they're always followed by something else. It was very difficult to find single species groups in particularly in that season. Maybe there are other seasons, and I studied largely in winters, starting November to about March, April. There are maybe other seasons where birds have, other foraging strategies, but in the winters you almost always see gregarious species being followed.
All right. So the next question is, how do the flocks ensure that they stay together?
How do they ensure that flocks stay together? Yeah, that's a difficult question to answer, but there are several different characteristics that probably lead to the flock staying together. Well, vocalization could be one of those. Species being or individuals being present in large numbers could be another. Species being able to detect and follow other species could be another. So all of these factors perhaps act in you know coincide with each other to be able to make sure that the flock stays together.
Interesting. So, do the nuclear species communicate with other species and form a flock? I mean,
Right. So the nuclear species do tend to be vocal. I can see that people are catching on with nuclear species and their characteristics and traits, which is why I was giving sort of disclaimers about the use of the terminology nuclear species. But nuclear species tend to be very vocal, and they do have well-developed alarm calling systems, like I was saying, because they are family groups of their own. So there may be some kid-related dynamic going on there. But whether they form a flock is something that is questionable, right? They are vocalizing, and there is some amount of public information that is generated from the vocalization of these species, and other species are attracted to this vocalization. So do nuclear species vocalize and communicate? Yes. They generate public information in the process, and it is possible that other species are attracted towards these vocalizations.
okay, I'm not sure if this question has already been answered. I'll still ask away.
Sure.
The intraspecifically gregarious species in a flock, what are the benefits when they lead the flock, and how are these benefits different from the other species within the flock?
Right. I think a part of this was covered when I was answering the question related to what benefits do intraspecifically gregarious species get. But I'm trying to read this question again. What are the benefits associated with gregarious species in the flock as they lead the flock? How these benefits different with other species? Okay. So I think Gayatri may be asking, what benefits do gregarious species provide, and how are they different from other species? So Gregary, like I was saying, gregarious species are known to be flock leaders. They are found in the front of the flock. They also tend to be often species that are very active and that tend to flush out other insects, that tend to disturb other insects, which may be caught by the species that are following. And these benefits may not, they're also known to vocalize and alarm call. So these benefits are not exclusive, or they're not extremely different, but not all. So for example, from saline species or sentinel species, there are very specific benefits. It's mainly protection benefits, whereas with gregarious species, you'll see both protection and foraging benefits that other species may get.
Okay, and Akshita is asking for more papers, but
Okay.
Apart from the ones that are already shared, could you like give a easier way?
I can share a list of species. I'm saying species papers with the NPTEL group with Jobin and Devika perhaps, and they can send it to you.
Okay. So, okay, this next question. First of all, first question is basically, is there a carrying capacity within a flock for competing species? Like, is there like a limit to the number of individuals? The other question is, so if the foraging technique is different, is the species with the same technique of foraging don't form? Okay.
I can't
No, I'm sorry. I was reading the question. Slightly. Yeah.
Okay.
So basically, what Krishna is trying to ask is, would a species with the same technique of foraging, are they able to form? Are they able to form like be a part within the same flock? Like, do the species have to have different foraging
different techniques? Yeah. Right. Yeah. Interesting. So about carrying capacity, That is an interesting question. You see a range of flock sizes, like I described, in and there's a range of, like, there's a lot of variation in what sizes of flocks you see, and a carrying capacity would be with respect to the environment, right? It would be with respect to a limited resource. Here it is difficult to evaluate what resource we are talking about, or what the limits of the resource that we are talking about are. So it's difficult to define a carrying capacity of a flock. Now, having said that, like I described, in the Western Ghats, we've seen up to 20 to 25 species participate in flocks, and it's rare to see more than that. That could also depend on the species pool itself. So how the community, how speciose the community of birds in that area is itself. So yeah, I think the question about carrying capacity is slightly more tricky. In the neotropics, where the pool of species, or even in Northeast India for that matter, where the pool of species itself is so diverse, you will see higher species numbers that participate in flocks. So it may have something to do more with the pool of species rather than the carrying capacity. So yeah, that is one point. You do see birds that use the same foraging technique in flocks. And like I was saying before, insects are a more dispersed resource. So it's not that conflict incidences don't arise. They do. There may be some conflict for the same insect sometimes here and there. There may be some chases in flocks. But largely, it seems like an association that works. You will see two species that have the same foraging technique participate in flocks at the same time. Then they may separate by canopy strata, or they may be one may be eating slightly larger insects than the other. So there may be subtle differences, and it's hard to say every single time. But it's important to think about it carefully. Even if they're using the same foraging technique, what is it that is different about these two species?
Okay, this next question.
It may not always, sorry, I'm just finished. It may not always be different.
Yeah. So this next question is quite interesting. I mean, are there freeloaders in a part? I mean, they basically wouldn't have any advantage to the flock, but they're still there, a part of a flock. So are there?
Yeah. Right. Yeah. Excellent. So yeah, definitely. I think that it is very likely that species don't contribute actively to the benefits in the flock. That is an interesting question. But there are things like, for example, species, when it joins the flock, by default, it's increasing the group size of the flock, right? It's contributing to the group size of the flock. So that way, it's contributing to the dilution effect. But it may so be that the species does not contribute so much with respect to food finding, or with respect to disturbing insects which become available to other birds. And the bird, however, on the other hand, the bird itself, is that individual itself is kind of benefiting from these feeding related benefits itself. So yeah, there can be free flocks.
Okay. So Anand Gopal is asking if there are some changes in the vocalization of birds when they are within a mixed species flock.
Oh, that's a very, very interesting question. It has not been studied so much, vocalization, changes in vocalization in flocks and outside of flocks. In fact, there is a student who is interested and is studying some vocal behavior in general. Actually, vocal behavior in mixed flocks is very understudied. So we would love to understand more about how vocalizations are different in flocks. But yeah, it's a question that's largely not been answered so far.
Okay, the next question is probably, I mean, contributing to the question already been asked. So MJ Singh is trying to say if many eyes hypothesis could actually, many eyes could also contribute to more successful foraging. Hence, can it be an independently stated mechanism?
Many eyes could be contributed to successful.
Like independent as in from the early warning hypothesis.
Right. We did. I mean, I think I stated it as an independent mechanism. Yes. But you're right that many eyes could also contribute to successful foraging. Many eyes also contributes to detection of predators. So it's related to two different things, like more individuals searching for insects, more individuals watching out for predators, and then early warning is separate from this. Yeah, you're right. I think they're trying to answer the question that was asked earlier. Yeah, you're right, John.
I mean, Divyapal is thanking you for your excellent presentation, and it was lucid and comprehensive.
Thank you.
So I think I misread Krishna's question. I think she has rephrased it over here. She's asking, do different species consciously communicate and form a flock?
Right. Yeah, thanks, Krishna. I think it's difficult to say whether species are communicating consciously, but what I wanted to say earlier, what I was trying to say earlier was that when a species vocalizes, it generates information which is then used by other species. So I'm still sticking with that answer. I do not know whether this is a conscious, it's difficult to answer these questions about whether a species is doing it consciously or not consciously, and is starting a flock. It is, yeah, I don't know. Umesh, what do you think about species starting its flocks consciously?
Well, like you say, Priti, it's difficult to tell whether the species is vocalizing consciously to start a flock, or whether it's some kind of ingrained, evolutionarily selected behavior. But I think your answer is right. It's that every vocalization carries some information, and therefore, I would stick with that answer too.
Okay. Thanks.
Okay, yeah. So, very last question for you, I think, Priti. I don't think there are more questions after that. But very similar to vocalization, are there different behaviors as well when the species is within a flock and when it's not?
Right. Yeah, that's also an interesting question, whether species behave differently when they are in flocks versus when they are not. And this behavior could be in terms of various things. It could be in terms of vocalization, it could be in terms of foraging strategy. And it is possible that species use slightly different feeding strategies when they are outside of flocks and inside mixed species flocks. But I can't off the top of my head think of any comparative work that has been done and demonstrated this, but it is definitely an interesting question. Like I was saying, sometimes when you're especially flock researchers, when they go out into the field in the peak flocking season, you almost always see species in flocks. So it's very difficult to record them outside of flocks, but that would make a really excellent study question. And if somebody was able to go out into the field and collect that data, it is likely that you'll find something very interesting.
Yeah. So Mrinal is also thanking you for great presentation and discussion, which is actually great. Thanks, Priti. So, okay, there is next question has come. Sorry, Priti, I have to still wait for a while. So that also still. Okay. So basically, MJ is talking about kleptoparasitism. I think literature that brings out that drongos also steal food from other species by mimicry. So doesn't that generate mistrust among other participating species towards the small birds, towards the same birds acting as sentinels?
Right. So mistrust is a very value judgment-laden term. So I wouldn't say mistrust. I would refrain from using mistrust. But yeah, so the drongos are known to steal food from mimicry, and many of you may have seen the videos that David Attenborough's documentary has, right? The drongos giving alarm calls and the meerkats are feeding. And the drongo gives out a warning, and there is a predator, and the meerkats is scurrying away to hide. And the first time it's a true warning, but then the drongo does a false alarm, and then it mimics a meerkat alarm call, and so on. So yeah, drongos are seen to do such interesting behaviors in different systems. And it is about really how many times the target species loses the food that it has captured, right? So there is of course a trade-off between what benefit species gets from the drongo alarm calls versus how much cost it pays in terms of the loss of food. And if the drongo alarm calls are so important, crucial to the survival of that particular individual, then it's a sort of much versus life kind of a dilemma, right? You would let go of lunch to save your own life. So it's an interesting dilemma to think about. It's definitely a trade-off that species probably think, in some ways have an understanding of. Yeah, I don't know if I've completely answered that question.
Okay, I think a lot of thank yous coming, I think, for you to read in the chat. So I'll move to Umesh's question. Umesh, would that be okay?
Sure, Jobin, go ahead. Thank you.
Yeah, so there was one question that came, no, there are actually two questions, same person. Okay, so I'll read out the question. I haven't read the question, but so my doubt was about the question regarding hornbills feeding off of 35 fruit trees in the absence of the competitor species. Does ecological relief indicate both population increase and niche expansion due to absence of competitor? Or and will niche expansion feeding from more trees always result in increased population size? Sorry, Umesh, do you want me to? I'll just copy this question on the chat box so that.
Yes, could you copy that question into the chat box for me, please? So about character displacement.
Sorry, Umesh, just one thing. The assignment is still open and the deadline is not passed yet. So, yeah, just want to make
Okay,
remind
so
you.
I'm not going to reveal anything about the assignment question, but about the character displacement, it's not necessarily only to do with morphology. For example, it's not necessarily that it's only beak size that changes or only body size that changes in the presence and the absence of a competitor. It could actually be changes in behavior. For example, if you have a certain species foraging only in the understory of a forest when there is a competitor in the canopy, and then when you remove the competitor species from the canopy, you have this other species expanding its range from the understory to the canopy as well. So, even foraging behaviors like that can change in response to the presence or the absence of a competitor species. So this is assignment 7, which apparently had been closed yesterday. I don't know what this question is.
No, it's not. Sorry, it's not a question. I think you've answered the question that's supposed to be answered.
Okay.
Yeah. So sorry, you want to say something? Sorry.
No, go ahead. I'll take the next question.
Yeah. So the next question is from the discussion forum. Okay, so the person is trying to ask how accurately can we age a bird once it has attained adult plumage?
It depends on which kinds of species you're talking about. Oh, once it's once it's got adult plumage.
Yeah.
In the hand, it's very, very difficult to judge what the age of a bird is. For all passerine species, it's actually impossible to do just by looking at the bird in the hand when it's an adult plumage. So you don't know whether it's 1 year old or 2 years old or 6 years old. There are ways to do this through molecular techniques. If you get a blood sample, then people have looked at the length of chromosomes, length of what are called telomeres in the chromosomes, and that's been used to figure out the age of species. But in the hand, it's near impossible once a species has got adult plumage.
Okay, so the next question is from your PPT, where you were, I think the slide was called Estimating Abundance through Mark Recapture. So the question is, there were numbers over there. So in this case, the 36 is an estimation and 40 is the actual population. Like it's a question mark, basically.
Yes, 36 is the estimate of the population size. The actual population size, which we actually don't know, right? We don't know what the actual population size is. We're trying to estimate that size. So the actual population size in nature, which we don't know, is in this case 40. So this is a sort of a contrived example. So we know that We know that there are 40 individuals in that habitat. If you have satisfied all the assumptions of the mark recapture model and so on, then the estimate that you get of population size will be very close to the actual true population size. And I didn't go over this in the presentation itself, but you also get an estimate of standard error. You get an estimate of the confidence that you have in your estimated population size. So you might get, for example, the mean estimated population size is 36. The range of population estimate could be from 32 to 40. So you'll also get sort of a band of, it's 95% likely that the population size is between 32 and 40.
All right. So Tahir is asking if is speciation happening by competition or because of competition? Is speciation happening by competition?
Is speciation happening by competition?
It's in the chat box. Yeah, these are the words.
Yeah, that's Tahir Ahmed's question. So there are several forms of speciation. Speciation by competition. Usually what happens is that, that there is this principle of competitive exclusion, which is very, very foundational in ecology, which states that two species that have exactly the same niche are cannot coexist. One will drive the other extinct. So two species that are perfect competitors, theory predicts that one of them is going to win and is going to drive the other one to extinction. But species that do compete with each other, you do see things like the ghost of competition past, where you see patterns, let's say, in foraging behavior, where you have an ancestral species that then splits up into multiple species. But usually the cause of that is, isolation, what's called vicariance. But I think there will be a session on speciation, and Robin will talk about, biogeography, and there might be more insights about the role of competition in speciation when that session happens.
All right. I think the next question is, I think it's related to your spot mapping slide. So
I see that. Is that one in the chat box?
Yeah. The one in the chat box.
So the question is call count specific to some birds as the other counts are not possible, or is there any other way to count? What is the accuracy of the count based on the fact that all the birds might not call when we are counting, and how do we identify same bird or different birds calling? If only male birds call, then how do we make the count inclusive of females? Right, that's a great question, and it actually shows some of the limitations of call counts. Right, so for example, if you want to estimate the abundance of francolins in the breeding season, and you know that these francolins are going to be calling, and they're going to have territories in the habitat, so you can actually identify from where each male is calling, and you know that this is one male in this location, that's another male in another location. If you know all of this, and you know that you're not confusing two males, then you can say the estimate of the population size with of males in this location is so and so. But of course, you don't know how many females are there. So it's true that the spot mapping technique is not suited generally for estimating bird population sizes. It's very, very specific cases where you know a lot about the biology of the species, and you know which individual, are males calling, are females calling, and so on and so forth. That spot mapping is useful as a technique.
Okay, I think the bird in your last slide, someone wants to know the ID of that bird. I think it was that can be answered later. So the next question is, what's the actual difference between exploitation and interference competition? And yeah, do the both of these? Yeah.
So the difference between exploitation and diffusion competition, you're absolutely right that both will decrease the fitness of the organism. But the difference between the two is the mechanism through which the competition acts. So interference competition is where there is a direct interaction between two competitor species. So I could, if I am species A and you are species B, then as competitors, I could actually chase you away from a resource. So for example, if I'm a hornbill, a large species foraging in a fig tree, and a bunch of green pigeons comes in to forage on the same figs, then I can directly, as a larger bodied species, I can directly chase away smaller species, for example, so that they are not competing for the same resources directly. So that's interference competition. There's a direct interference of one species interfering with the other. In exploitation competition, these two species might not ever meet. They might not ever have any aggression between them. So, but they're feeding on the same resource. So, for example, fig trees in the daytime are fed on by barbets and pigeons and so on. In the night, they're fed on by bats. Now, if in the morning there's a lot of foraging happening, the number of figs comes down, and there's not that much resource available for the bats. The bats and the barbets are never going to meet, but they're never going to interact. But because they're feeding on the same resource, if one feeds depletes resources, it is going to affect the fitness of the other, even though they never meet. So that is exploitation competition. It's also called scramble competition, which is the exploitation of a common resource.
Okay, so this other question is about Transect study, and while doing Transect, is it not difficult to actually count birds, like come to a particular number? And yeah, I mean, wouldn't the counting give us a valid non-bias? Would it give us a valid non-biased data when the counting itself is so tough and cumbersome? Without missing, yeah.
It takes a lot of practice. It's not easy when you start doing these things. It takes a lot of practice to be able to quickly calculate or use the compass and the rangefinder to get at these values. But it's not impossible. And the whole point of doing this is to estimate how many birds you're missing, right? So, for example, if it's ground birds, and you know that ground birds are difficult to see, then the probability of detection of these ground birds is going to be low, correct? So the fact that you might miss birds because there are too many birds in the habitat, and all of this is happening at the same time, is also accounted for by that detection probability process. So the birds that are really tough to see in the forests will have a low detection probability, and the birds that are easy to see in the forest will have a high detection probability. So we don't calculate the detection probability for all bird species together. You ideally want a separate detection probability for each species, because the behavior of each species and the ability to see or hear each species differs from species to species. So even if you miss birds because there are too many, if you have enough data, you will be able to estimate how many birds you have missed. Jobin, I'll just take that follow-up question there, which is that in a given patch of forest, one draws the transect line and then counts the birds in the ESWs. How does one cover patches of forest that do not cover the assumed rectangular patch and will not be the exact dimensions of the ESWs? Right? So we don't know what the ESW is when we're doing a transect. You're walking along a transect, you don't know what the effective strip width is. This is not something that we know beforehand. The effective strip width is calculated or estimated after we come back and analyze the data. So birds are being counted both within what will ultimately be the effective strip width and outside the effective strip width. It is the rate at which detection declines with distance from the transect that gives you the effective strip width. That can vary from transect to transect. It can vary from species to species. It can vary from habitat to habitat. It can vary from season to season. So the effective strip width is a variable measure. We don't know what that measure is. It's estimated from the relationship between distance from the transect and detection probability.
Okay. Okay, so the next question is in a census counting, are teams given multiple patches of, say, a rectangular forest, and then the sum total of all the counts become the total estimated population for a given species?
Yes, that's one way to do a census, where you split up, you divide the habitat that you're trying to sample, that you're trying to census, in this case forest. You divide that up into, you know, blocks or rectangular sections, and then each team is assigned one section, and then, you know, they come back and total up the numbers, and that's how you get a
So the next question is, do scientists also use combination of line transect and point count to get a total estimate, like a combination?
So you can't, analytically, you can't combine lines and points. When you come back and do the analysis, you cannot combine lines and points. What you could do is to sample the same habitat using line transect and using point counts, but the data that you get will have to be handled separately. And the estimates that you get will be separate for point counts and line transect. So it's not possible to combine both of them into one analytical framework.
All right. So the next question is about how to estimate populations when in water bodies, like basically for water birds, because it has to be, I mean, does it have to be point count? Because we cannot seem to do a transect study in water bodies. And also, how do we count mixed flocks? Like, how do we estimate population of mixed flocks?
So, you know, water birds typically are censused by direct counts, you know, because the body of water is open. You can stand on the shore with a scope, and you can actually count the number of birds that are there on that body of water. And you're unlikely to miss species because there's not much vegetation in that area. Of course, it depends on the skill of the person who's doing this and so on. It takes practice to get accurate counts from direct counts of ducks and so on. But that's the typical way in which these are counted. Now, if you have a water body that has vegetation on it, let's say, you know, and the vegetation could hide species, like in a forest, you can actually do a line transit from a boat. And, you know, line transects have been used to in water to estimate densities of things like, you know, whales and dolphins and so on. And the same principle can be applied to birds, to water birds. So you can actually take a boat and, you know, go in a straight line through the water body and count birds on both sides and count the distances to these birds on both sides, and actually use line transit methodology and line transit analysis to get at population sizes or population densities of birds in water. So if the birds in the water are swimming around, how do we count them? Of course, yes, that the assumption of the line transit, of course, is that the speed at which the observer is moving is much faster than the speed at which the birds are moving. So if the birds are swimming around really fast and the boat is going at a very slow speed, then the line transit methodology is not appropriate. It's just not the way to count fast moving species. How do we count mixed flocks? So when you're walking along a line transect, what we do when we encounter a flock is to take the distance to the center of the flock and note it down as a mixed species flock. And you got, you know, 7 species. They were all foraging, you know, 20 meters away at an angle of 20 degrees. And all of them will get the same bearing and the same distance. You're taking the bearing and the distance to the center of the mixed flock. Deepika's question is, how do you avoid duplicate counting of the same individuals as birds keep flying around? So Deepika, like I was saying, you know, the line transect methodology is only going to work in a situation where the birds are moving relatively slower than the observer on the line transect. The other thing to consider when you're doing line transects is that obviously you're not going to do only one line in a habitat, you're going to do multiple lines in a habitat, and you have to make sure that the distance between the lines is a minimum distance such that the same bird cannot be counted from both lines. So when you're doing a line transect study, you can't have the home range of one bird overlapping two lines, because then you're going to count the bird here and you're going to count the bird here. So that's a double counting. So the birds have to be moving relatively slowly while you're walking the line, and the next line that you're sampling should be far enough that the same bird is not counted on both the lines. Now, obviously, that distance is going to vary from species to species, right? Some species are going to have very large home ranges. So if you're counting, you know, something like, you know, eagles, you know, I'm just giving you an example here. You know, nobody really counts eagles using line transects. But if you're counting a species that has very large home ranges, then the transects also have to be placed very far apart. If you're counting a species that has a small home range, then the transects can be placed relatively closer to each other. So it depends on all of these considerations to avoid double counting. Amit, sorry, Sharda has a question. For a large flock of birds, how is counting done since one can't count all? So you can't count all for a large flock of birds. You're going to miss some. So it, again, you know, the large flock of birds could be water birds on a water body. It could be, you know, rosy starlings in a grassland or an agricultural habitat. And the methods to count these are going to vary. So, you know, for example, starlings or miners are large birds that have large flocks, and they come back to roost in the same location every day. So in that case, you would do a roost count. In the case of water birds, you might do a direct count. So it's going to vary with from species to species. The precise method that you use to count the number of individuals in the flock is going to differ. Amit Shankar has a question: is the documentation and methodology available on bird count available online, or can this documentation process be shared, please? This is all available online. There is, I'm just going to type this into the chat box. There is a website called FIDOT.org, which deals with the market capture methodology. There's a manual that you can download online for free, and it explains the whole market capture methodology very nicely. The line transect methodology also is available online. You can find that. I'll just paste the link here. You can find that in here. Here is one way to do, one place where you can find this information. But a lot of this information is completely free and available online. If you just Google ways, you know, line transect methodology, or you Google capture market capture, you'll be able to find this. During which season, Anuprita has a question asking, during which season are birds counted? How frequently are they counted per year? That depends, Anuprita, on the kind of study that you're interested in doing. This will vary from study to study. So if you're interested in looking at breeding bird densities, you would count them in the breeding season. If you're interested in looking at wintering bird densities, you would count them in the winter. How frequently are they counted? That again depends on what you're trying to do with your study. So it's variable from study to study. So if I just want to monitor how breeding bird populations are changing from year to year, then I will, you know, go out and measure only breeding bird populations year after year. If I'm interested in looking at, for example, survival of birds between summer and winter, and the survival of birds between the winter to summer, then I will sample in summer, I will sample in winter, I will sample the next summer and the next winter. So twice a year. So it depends really on the kind of question that you're interested in. You might even ask, what is the day-to-day variation in the densities of species? That would mean you would actually go out and measure these things. You could actually go out and measure these things every day. So it depends on what kind of study that you are conducting. There's no hard and fast rule for when birds should be counted. How do we calculate the home range of a species? Does it vary depending on the habitat for the same species? So calculating the home range of a species is quite difficult, especially for small birds, because the ideal way in which home ranges are calculated is where you fit a radio transmitter or a GPS tag onto an animal, and you get records of the locations where this animal was, and then those locations are then used to calculate the home range of the bird or of an animal. So this has been done for animals like elephants and tigers and deer and so on, which are large and which can actually carry these collars or tags. With birds, this has been done for larger raptors like eagles and vultures and condors. Fitting a GPS or a satellite tag onto the backs of these birds to figure out what the habitat range of the species is. For smaller passerines, the problem is that they're too small to carry a tag. So estimating home ranges of these birds is more difficult. One of the ways in which it has been done in the past is spot mapping. But again, spot mapping can provide unreliable results if all the assumptions of spot mapping are not met. Habitat's home range size does vary for the same species from habitat to habitat. So if you often depends, for example, on the distribution of resources. So if you have a habitat where there are lots of resources, the density of resources is high, then an individual needs a much smaller area to be able to satisfy its energy requirements. Whereas in another habitat where the resources are less dense, they're more sparse, the amount of resources that need to be gathered to satisfy energy requirements will have to come from a larger area. So it does depend on habitat to habitat through the distribution of resources, what exactly the home range size of this species would be. Krishna has another question. So this is about point counts. So as bird count will be lower in a 10-meter radius, what is the optimum radius to be chosen for point count? Is there some method based on the density of land cover, like in a forest or grassland, based on the visibility, or accepted norm based on vision maximum birds can be located with minimum error? So Krishna, again, in the classical point count method, you're not actually counting birds within a certain radius. You're not setting any particular radius within which birds have to be counted. What you're doing is you're measuring the radial distance to every bird, and then you're coming back and calculating the effective radial distance. So there is no hard and fast set rule for distance sampling based on a line transect or a point count within which, within this distance is how I'm going to count birds. Now, of course, if you have a fixed radius point count, then it depends from habitat to habitat. It depends from species to species. So for instance, in forest, it would depend on, for example, if I would have to have some very clear reason to say, let's say that all birds within 50 meters of myself, I'm not going to miss. Now that distance might extend to 100 meters in a grassland because visibility is higher. So it in a fixed radius point count or a belt transect, which is a fixed width line transect, the distance within which you assume that no birds have been missed by the observer depends on visibility in a habitat. It depends on what kind of species we're talking about. Is the species very vocal? Is the species very, spectacular, easy to see, easy to hear? Then the radial width might be higher. If the species is very cryptic and you're likely to detect it only very, very close to you, then that radius has to become smaller because you're not likely to detect that species beyond a certain distance. So there's no industry standard or accepted norm. It varies from species to species, varies from habitat to habitat. Point count is actually usually used with binoculars. Anand's question is using a sampling count, and since birds are mobile, is there not a chance of recording sighting the same bird multiple times? I think we've already gone over this. Line transect or point count methodology is only appropriate for birds that are relatively slow moving compared to the observer. And two, the distance between the lines and the distance between the points has to be minimum such that you're not double counting, you're not counting the same individuals on neighboring lines or neighboring points. So the distance between the point counts or the distance between the line transects has to be enough that the home range of one individual does not overlap two lines or 2 points. So that's important for line transect. Can the camera be used? Camera image be added? Usually, you know, I am familiar with forests and doing line transits and point counts in forests. Usually, you know, when you go into the forest, there's quite a lot happening. And if you're actually doing a proper line transect or a proper point count, we usually don't do any photography. And that's because while doing photography, you might miss, while taking photographs, you might miss individuals of species. And we ideally don't want to miss as much as possible. We want to have detection probability as high as possible so that our estimates of abundance are better. You know, the lower the detection probability, the lower the confidence we have in the estimates of density, species density or species abundance. So, you know, when you're taking photographs, you're going to miss other individuals in the habitat. So I at least have never used the camera on a line transit or a point count. So, Jobin, are there any other questions?
No, I mean, I think we have covered the discussion forum questions and the questions on the chat box as well.
Great.
So, yeah.
Then,
Thanks.
well,
Thanks, Umesh.
thank you very much for joining. I hope you enjoyed those sessions on estimating bird populations and you know, measuring bird populations and communities. Thank you.
Yeah, thanks, Umesh. Thanks,
Thanks,
Priti, for a great session. And thanks, everyone, for joining in. Thanks a lot.
Thank you. Bye.